Check it out now, y'all.
nanoHUB-U, online instruction.
All right, welcome back. Now we have spent almost 4.5 weeks on thinking about various aspects of nanobiosensors and what makes it special, right? And we have talked about lots of fundamental limits also. There was settling time limit for diffusion, and we have talked about the salt screening limit, and then we have talked about the selectivity issues. Some of them are very fundamental. And so the question you might always ask that if there are so many troubles, then is even nanobiosensors going to go anywhere? The answer is that not only can it go somewhere, it has already have had impressive performance. So I'm very excited today to tell you, bring all these pieces together. In the next 2 lectures, I'll tell you about a genome sequencer. Genome sequencer is essentially a machine that reads the letters of the DNA, the A, T, C, G, the sequence of letters in the DNA, and can tell one that whether there is a genetic predisposition to some disease or not, and in general provide a wealth of information about a person. So let's get started. My idea here is not to get the technology right per se, but to show you how all things that we have learned so far can come together in a beautiful integrated whole. So I'll first explain to you that how does genome sequencing work? How do you read a book that has 4 billion letters, sort of, more than, let's say, 3 billion letters? How do you read a book like that? So I'll start by explaining the basics of this. I hope you'll find it very interesting. And then I will tell you that although most of the time it has been traditionally done by optical methods, these electrical nanobiosensors, these potentiometric sensors that we have talked about, how they are making considerable inroads. So I'll tell you a little bit about that, and then conclude by saying that nowadays people are putting hundreds of millions of nanobiosensors in parallel so that you can read the whole thing in a very short, the whole genome in a very short period of time. So recall that in the last lecture, we talked about a cell being very complicated, and we lysed the cell, right? Broke the cell apart, and then there were all sorts of proteins, and the proteins themselves we separated by centrifuge, by rotating it at very high speed. And at the end, we were able to separate the chromosomes and the DNA that are embedded in it. And by now you know that the DNA forms a double helix. There are these hydrogen bonds which keeps them together. You have to have salt so that they don't fly apart because of the negative charges associated with it. But the bottom line is this sequence of A, T, C, G, how they are written, is essentially the microcode for, if we think ourselves as a microprocessor, this is what the microcode of that microprocessor is. Now it turns out that the genome is generally, this microcode depends on the organism. For example, an HIV virus would have only 10,000. It's a very small genome. And E. coli, sometimes we have E. coli outbreak that has about 4,600,000 cells, but a human being has far more, a couple of billion. And so when we talk about genome sequencing, what we are talking about is being able to read the sequence and see what the book says. Now you might say, why should we care? Doesn't everybody's genome exactly the same? Not really. Everybody's eye color is different. Everybody looks different, slightly different height, build, and all those things. Is because everybody's personal genome book is written slightly differently. And that is what gets translated into our being, every human being different. And that's very important, because if you knew what the genomic makeup of a person is, one can design a medicine that is specifically tailored for a person. And this is called a personalized medicine, so that the Tylenol they make for you is not for your friend or somebody else. It is tailored exactly for your genetic makeup, your protein network. So that is what is the promise of this field, that if you can read genome, then you will be able to do these things. So people had been trying to do it for a while, you may know. And even 20 years ago, if you had to read it, there were these big machines, football-sized fields, where these were being done. People are trying to read the DNA bases this way, very big. And if you had to compare, it will be like a computer. It looks like a computer from 100 years old, a 100 years old mechanical computer. You see what has happened, that recently this whole system has been replaced by a little microchip. Whatever these big machines could do, the microchip can do the same thing. And the salient feature of these microchips are these little wells, this honeycomb. There are millions and millions of these honeycomb ones. And here you see the side view. So these are the wells where the DNA molecule will be stored. And each one will have slightly different DNA molecule. And I'll explain where they're coming from. And although you cannot probably see it very well, here are the transistors. So we'll be using potentiometric sensors in order to read the DNA. I'll explain how that goes. And if you had to compare the progress in last 20 years, that will be like having the same progress of last 100 years or 150 years associated with microchip. Why this extraordinary progress? Is because nanotechnology is sort of melding with the biotechnology for the first time. And very exciting things are happening that no one thought would be possible even 10 years ago. So I'll explain how everything that we have learned essentially can be used to understand this technology. And again, just to give you some perspective, that these days a genome sequence machine looks almost like a computer. Just like you have a chip inside, there is a chip inside which does the sequencing. And inside here you have individual transistors, for example. This is a high-K metal gate from a few years ago, and the corresponding technology is exactly the same. So below this honeycomb type lattices, there is this underlying transistor technology is exactly borrowed from classical standard nanotechnology of transistors. So therefore, since you can build on top of it, the chemistry can take all the advantages of modern nanotechnology. So that has given this impressive power for this technology to make such a quick progress in such a short period of time. Okay, so let me explain now how it works. I'll walk you through slowly. Start with the DNA and assume it's 4 billion base pair long. Start with it and let's cut it. There is a special enzyme which cuts it into, first of all, we make it single-stranded by increasing the temperature to 92 degrees. It will get split because the bonding can no longer hold. And then let's cut it into pieces of 100 to 1,000 base pairs. So little books. So you cut it and then, so each sequence is different, as you realize. The magenta, the green and all, they are slightly different things, but each on the order of about 100 to 1,000. So it's like you have a big book, you're tearing off 100-page segments because you'll be giving it to different person to read it. And there'll be 100 million people who will be reading the book in parallel. That gives it the power. All right. Then what you do this copy. Or amplify. How do you do it? You already know it. In the last lecture, we talked about PCR amplification. So what I just showed you here is that you have to do a PCR amplification a couple of times, let's say. Generally, you will have 10,000 copies. 10,000, you will photocopy 10,000 times by this PCR. All right. So now you attach it to the substrate like this. Remember, each one of the green, the whole thing looks green, but there is really 100 different letters or 100 different base pairs along the green. Similarly, the blue, all the blue look the same because they have the same sequence, different from the green. But within each green, there are 100 different letters. Now let's take one of those. And let's say it has A, T, C, G. I have used a different color. Of course, they don't have any color, it's just different sequences. Now, let's bring a solution flow the solution on top of it. So let's say there is a polymerase. Polymerase is something, it's like an enzyme which allows a reaction to take place. And in the solution, let's say you bring in a bunch of ATCG. Now only T will bind. And if the T binds, then again you bring in the 4 solutions. And there will be this copy step by step. That is what is called sequence by synthesis. So you essentially build this copy one thing at a time. And every time you build it, it turns out that if you use a spatial tag, let's say this is a red tag, then every time you build it, there'll be a red color associated with this one. And that tells you that since it is A, the corresponding conjugate later must have been T. So here, as you are going up along the stack, you will be reading, the color will be changing as it's going from T, A, C, G, and so on and so forth. And therefore, from this color, you can take continuously, every time you flow in the fluid, you'll be taking a picture. And so let's say in the beginning, you have these 4 wells, 4 segments. First, you bring in the solutions. And then before you bring in the solution, everything is dark. But let's say the first one was in the bottom of this brown one was A. And so it will have a red color. The other one had a G, C, and T. So if you take an image, you will see these 4 colors. Then you bring the solution next time. Then layer by layer by layer, you will be gradually be reading these structures. And essentially, once you are done with 100 layers, since the whole stack is 100, then therefore you can read up the entire sequence that way. And in fact, this is one of the real pictures. So you can see there are yellow, red, and green saying that in this layer, so you're going from 0 to 100. Let's say this is for the 43rd. So on the 43rd floor, you can sort of see that this one has a green, and the green is a G. That means that you have a corresponding C at that stack. And so each one of them is a well. We just showed you 4, but in principle, there can be hundreds of thousands of this in parallel. So you take this picture layer by layer by layer, and there will be how many pictures there will be? If it's 100 base pair long sequence, there will be 100 pictures. You analyze those pictures, put them together, and then you have sequenced the genome. All right. Now, one thing I want to emphasize that this requires tagging, this requires taking pictures, image analysis and all. It would be much better if we could do the whole thing electrically. So let's see whether we can do that. Remember our story about pH sensing by a transistor. You may remember that pH is essentially the log or potential of hydrogen in the log scale. And in neutral water, the pH was 7. And if it is acidic, then the pH became smaller. If it is basic, then or alkaline, the pH got larger, meaning that the concentration was smaller. And we were able to discuss from slide 13, from 13 to 18, lectures 13 to 18, that we have this OH group, and therefore when the hydrogen concentration changes, that changes the surface concentration, and therefore this change in the hydrogen concentration is reflected in the transistor current. And so that's something we already knew, right? We found that to be a parasitic effect. It was good for orange juice, but at any time we wanted to do the biomolecules, this was a bad thing. You may remember. If not, please go back and you can take a quick look. It turns out the bad thing can now be flipped on its head and used for biosensing. So I'll explain how that goes. And the bottom line is that as the hydrogen concentration is changing, the pH is changing, there will be shift in the current, and the corresponding shift in the current can be read off as a signature of the hydrogen concentration. All right. So how does it work then? So this is how it works. So what happens, and I will explain these things in more detail in the next lecture also, each one of the segments, what is done is that this is sort of put in the microbead. So you have a solution, you have in a lot of microbeads, and on each one of the microbead, you will have one segment of it attached to it. And all of the copies, 10,000 of them, are of the same type. How do you do that? I'll explain that later. But let's assume that every microbead has 10,000 copies of the same segment of the book or same chapter of the book. This one has the green chapter, this one has the magenta chapter, blue chapter, and so on and so forth. 10,000 photocopies of the same chapter sitting on the microbead. Then what you do, you remember the little hexagonal holes? You randomly distribute the microbeads so that they get inserted in the holes. Now, if they get inserted in the holes, remember, this one has a green chapter, the other one has a blue chapter. So everyone will have a slightly different chapter of the whole book. Remember, there are hundreds of millions of them. So therefore, you will have 100 million chapters of the book sitting in these wells. All right. Now, once you have it, let's say you wash in the molecule A, one of the bases of molecule A. And if within this segment there is A, then it will start the conjugation process. And as a result, let's say this is the bead, the molecules are the DNA molecules not shown here in this yellow bead, but they are assumed. And as soon as you bring in this A molecule, then if there is a conjugation, it will release a hydrogen. Because every time there is this conjugation of the molecules, it releases a proton as a byproduct of the reaction. And this hydrogen, what it will do, the biomolecules will not diffuse. Rather, the proton will be the messenger that a conjugation has occurred. And so therefore, this biomolecule, this proton change will be reflected in the transistor, and therefore it will signal that a conjugation has occurred. And so how it works is that, let's say you have already built up to this far, and then you have the A molecule coming in that will conjugate with T, and as soon as it does so, it will release a hydrogen that will change the local pH of the individual wells. And when the local change in the pH is picked up by the silicon transistor underneath, we are in good shape. But remember, this will be a transient signal. Why? Because there will be some hydrogen produced. So 10,000 copies of the proton will be produced, but they will stay in, but this is an open well, and after a while, all the hydrogen will leave. And so the pH will initially jump once the molecules are getting incorporated, and eventually when the protons have left the well, it will go down to this original value. And then you can bring in the next molecule, and gradually build it up. And every time you have a pulse, it says a successful conjugation. And so the pH will go like that. And so it will shift and then shift back. And from this shift and shift back, you can read that the successful conjugation has occurred. Okay. So this is relatively simple. This is essentially amplifying the same point, that let's say this is a DNA sequence you want to read. This is the green chapter, let's say, that has 100 base pair. You have already built up to this point. You need a polymerase to make sure that the building doesn't start occurring anywhere else, but it starts occurring from the end of the previous build. And when the new molecule comes in, let's say a C comes in, and then only then it will react with G and hydrogen will be liberated. If the other things came in, for example, A, T, and G, as you are washing one after another, you see there will not be any reaction. And if there is no reaction, there will not be any signal, no proton generation. So therefore, by sequentially washing with these four and looking at what point you have a signal, essentially you can say what corresponding letter there was. And then you can step through this one at a time. And so these are, for example, some signals, typical signals that you can see in the system. For example, in this particular case, when the GCTA, so there is no corresponding conjugation for T. So there is no corresponding signal for this system. All right, so this is how you read. Now, how do you model such structures? Turns out that you can model it very easy. Here is the local generation of way of beating the diffusion limit. Do you remember that I said there are multiple ways of beating the diffusion limit? What was the first one? The first one was in terms of you can concentrate, or first one was biobarcode. That's one way of doing it. The second one was droplet-based evaporation. And the third one was local generation. You see, all the DNA molecules are so close to the sensor now that they don't have to swim anywhere. They are locally generated. And also proton are sort of being used as a messenger. And so therefore, the protons can move much, much faster than the DNA can. So therefore, this system essentially doesn't have the classical diffusion limit associated with big DNA. Of course, it has a diffusion limit associated with the proton, but they move very, very fast. So therefore, when you write the proton diffusion equation, the thing that we learned in lecture 4 to 12, you use that in order to know how long it takes for the protons to register on the sensor surface. Then you remember that we did all this electrostatic modeling. If you change the pH of the solution in the analyte, then how does it change the current in the silicon or silicon transistor? We did the electrostatics and all those things. You remember surface charge calculation and all those. Those will essentially be the next step. And you may also remember this complicated formula that we derived. And finally, you can then translate the corresponding current. Of course, one thing one has to always be very careful about, that this noise associated with the channel the signal has to be larger than the noise. I will derive the relationship. I haven't done so far. I'll derive the relationship on the supplementary materials. But the bottom line I wanted to point out was that it is inversely proportional to the area. So if you make a transistor very small, it becomes more noisy, right? And so therefore, in that case, your signal to noise ratio can degrade. So therefore, you cannot make this transistors arbitrarily small. You have to keep it some size so that the noise is small, so that you can detect the change in the signal. But essentially, there's nothing new. These are the things that we have discussed in potentiometric sensors and the diffusion limits. Okay, and in fact, if you do a simulation like this, it turns out that you can reproduce the experimental pulse very, very well. Remember, at time t equals 0, the base molecules are coming in, they are reacting with the sensor surface, releasing this host of protons in the solution. The protons are diffusing, and then they are coming to the sensor surface, and so that gives you the peak. And eventually, as the protons they diffuse out of the well, the signal goes down. And a very interesting thing, and you will do it as a part of your homework, is to show that if you scale everything by a factor of kappa, make the width smaller, make the well smaller, make the transistor or everything smaller by a factor of kappa, the peak does not change, the signal does not change, right? You have a fewer molecules for sure, because the bead is smaller now, because the whole thing is smaller, but the area of the number of molecules has been reduced by 4 pi r square, right, in that proportion. However, the well is smaller also, so therefore, as a result, your net density is such that the peak signal is essentially unchanged. So that's a very good thing, because it says that actually, even if you make the well smaller, it will not really matter, because in that case, the signal can essentially remain the same, and you can cram many, many more nanobiosensors into the same system. So let me summarize to see, because it's very important that you see how this technology actually beats several limits. One is, how did it beat the diffusion limit? Well, first of all, you cannot beat the diffusion limit. It is only that it used local generation so that L was small. Remember, it is L square over D. And second, it had a substitute diffuser. So DNA was very heavy. It cannot run as fast. So it hired somebody, the proton, which is lighter and more mobile, to do this job for it. And so that's why this substitute diffuser was very important to sort of beat the diffusion limit or sort of make the diffusion limit irrelevant. How did it improve sensitivity? Well, the way it improved sensitivity is making very, very small transistor, working with very small potentiometric sensors that has very high current and therefore and very thin gate region separating the fluid from the channel. And this thin gate essentially allowed it to couple to the channel, and it didn't have any corresponding screening limit per se, strong screening limit, because the protons were sitting just close to the sensor surface. And so therefore this long screening issue, a long DNA standing like this and getting screened out, none of these issues actually matter here. A very important point is that it also had a global, all these wells actually shared a single reference electrode. It doesn't have reference electrode in each one, the individual one. That will be too difficult to miniaturize. So these are the ways by which this sensitivity was improved. And what about selectivity? How was selectivity challenge addressed? Well, it was addressed by this PCR amplification. Remember, in the beginning, the DNA was segmented and each chapter was photocopied 10,000 times before being loaded onto the bead. And so the PCR amplification allowed significant enhancement in signal-to-noise ratio. And also these experiments are done multiple times. Each time it is cut at a slightly different location randomly. And then you read one time you read the whole book by assembling the pieces together. Next time you read the whole book by assembling the pieces together in a slightly different chapter width. And that way you can also significantly enhance selectivity. And finally, there are statistical techniques taken from information theory. I'll explain in the next lecture. That also enhances selectivity. So let me conclude then. The pH-based genome sequencer really addresses the fundamental problems of settling time, sensitivity, and selectivity of nanobiosensors. That's why the technology is successful. Now, integration with the nanotechnology, this IC fabrication, it's remarkable that the number of transistors in a single IC is these days a couple of billion, right? And so they are very, very small, of the size of a virus or less. And so therefore you can have hundreds of millions of these cells working in parallel to read the book. The book is 4 billion letters long. So this combination, this advances in the IC technology and advances in biotechnology, I will explain what emulsion PCR is in the next lecture, that these two fundamental development over the last 15 years allowed this development to take this significant advance to occur. Now, this issue about PCR amplification, that you had to photocopy 10,000 times before you could read them, is sort of really not good. And we needed 10,000 copies because it would have created 10,000 protons, which could be read. But you see, if my sensor was more sensitive, that it could work with fewer copies of protons or fewer smaller changes in pH, then I could get rid of this photocopy or PCR step altogether. And therefore, the various types of ways that I have said about enhancing sensitivity, well, you can use them eventually so that this big photocopying step, quote-unquote photocopying step, is no longer necessary, right? One thing I want to quickly mention, so this is one thing I didn't mention was this importance of this polymer. You see, why do we have a polymer? Generally, we have an oxide and the transistor underneath. Why do we have a polymer? It turns out that the DNA, these protons diffuse so fast that unless we slow it down, the electronics will not be able to read it. So not only it doesn't have a diffusion problem, it is coming so fast that my electronics is not fast enough to keep it. And you can see it's a fraction of a second, let's say a couple of 100 milliseconds, within which this comes. So therefore, the polymer layer is there to slow things down. So the proton moves a little slowly so that my electronics can sample the signal properly. And this is a very important thing that I forgot to include, but hopefully you will realize that this is very important so the system works as a whole. That's it. I will, in the next lecture, I will tell you a little bit more details about the problems associated with the technology and how those problems are addressed. But taken together, you can see how the pieces are coming together to make a technology successful. That's it. I'll see you next time.